Okay, our last little series of lectures here for Chapter 17 will involve a very important topic, and that is blood transfusions and blood type, which of course we've all heard of before, even if you don't know your blood type. All right, first of all, blood loss has to be treated very quickly. Blood volume has to be restored. Now, of course, the best way to do that is to give somebody a transfusion of blood from a donor, since it's better to replace lost blood with blood, because then that's going to have all the components that do all the nice things that we've been learning about here in Chapter 17. However, you can't always do that right away, and the most immediate concern from blood loss is potential death from shock, and this is actually due to low blood pressure, low BP. All right, so if you lose enough blood out of your blood vessels, your cardiovascular system is not going to be able to maintain enough blood pressure to circulate the blood throughout the body. If that happens, then the blood does not flow to your vital organs as quickly. Your poor little vital organs don't have enough oxygen gas and nutrients. They can't make their ATP, and cells die very quickly when they can't make their ATP. So that's the most immediate concern. When you don't have blood around right away for a transfusion, you've got to replace this volume immediately. And typically that's done with normal saline or an electrolyte solution, such as Ringer's solution or Ringer's lactate solution. And no, this does not have blood cells in it, but what it does is it mimics the plasma electrolyte composition. Normal saline is actually about 0.9% in chloride, and so the solute concentration in there is essentially the same as the overall solute concentration you would find in your blood and also your interstitial fluids. So this helps maintain the proper osmotic pressure of your extracellular fluids versus your intracellular fluids, which is very important. You can't just put pure water into somebody's bloodstream, because that's going to screw up solute concentrations and is going to cause problems with osmosis. So if you do that, as you get enough volume back into the bloodstream, that can help maintain blood pressure and can help prevent a patient from going into shock. Another thing that can be used are called plasma expanders. Okay, and what plasma expanders do, these are large molecules. So there are things like, now sometimes they may give a patient human serum albumin. Albumins are proteins that you normally find in the blood. Remember, those are one of the main components of your blood plasma. And those are large molecules. They stay in your blood vessels. They don't pass out of your blood vessels and into surrounding tissue fluids very well. And albumins attract water because they're solutes. So they help make sure that you are keeping enough water in your blood fluids and not losing that water to surrounding tissues. All right, so if you've lost blood, you've lost quite a bit of your albumin, and that can be replaced artificially with human serum albumin or hetastarch. That would be a large carbohydrate molecule. Dextran, same type of deal. That's a polysaccharide. And so if you put those into somebody's blood vessels, they will also attract water. They'll bind lots of water and help keep water in your blood vessels simply through osmosis. So these types of things mimic the osmotic properties of albumin. They help attract water and help keep your the water volume in your bloodstream up quite a bit. These plasma expanders, though, are using those more expensive, especially that human serum albumin is going to be very expensive. And you have to be very careful with these, because you don't want to wind up, you have to put in just the right amount. If you put in too much, then these things can attract so much fluid out of the surrounding tissues and into the bloodstream that you could actually wind up dehydrating a patient's tissue. So you have to be very very careful with those types of things with patients. All right, then ideally when you have a substantial amount of blood loss and it's not really going to be possible for a patient to regenerate the blood quickly enough, then you have to give blood transfusions. And those can include whole blood transfusions. And a whole blood transfusion is just what it sounds. You're actually getting whole blood, so plasma, red blood cells, and so forth. Or patients can sometimes get just packed red blood cells, and that's where the plasma has been removed and the white blood cells have been removed, and you're mostly just giving the patient that red blood cell component, because really the most critical thing to restore is that oxygen carrying capacity. If you don't have enough red blood cells, you're not going to be able to circulate enough oxygen gas to your tissues. Now, there is a problem. As we all know, you have to get the correct blood type. There are different blood types that exist. If you get the wrong type, it can actually kill you. So blood typing is something that is extremely important. All right, what do we mean by blood typing? All right, so if you look from person to person, there are a number of different types of molecules present on the surfaces of our red blood cells. And most of these are glycoproteins that are present on the surface. Those are protein-sugar combinations that are sitting in your cell membranes. You have glycolipids, sugar-lipid combinations that are sitting in your cell membranes. And if you look from person to person, most of those are going to be very similar or almost identical. However, there are a couple of types that do tend to differ from person to person, and those are called your ABO and your RH antigens. Antigens, what is an antigen? An antigen is a molecule that is recognized by your immune system. It literally means antibody generator. So when your immune system sees this particular molecule, it will generate weaponry to try to attack it. And those weapons include antibodies. Okay, antibodies are protein weapons that your immune system makes when it sees particular antigens or antibody generators. All right, antibodies typically have kind of a Y shape to them. They are proteins. They're not cells. They're protein molecules that kind of have a Y shape. And here what you're seeing are some Y-shaped antibodies that have attached to red blood cells. So they're sticking to antigens on the surfaces of those red blood cells. And if you notice, a typical antibody can attach to an antigen at this tip and then over here at this tip as well, because it has a Y shape. So because of that, antibodies can actually clump things together. And that's supposed to have an advantage to helping clump together things like bacteria that you get exposed to and make antibodies to, because that winds up helping you clear them out more quickly. But in the case of if you get a blood transfusion with the wrong type, those transfused blood cells can wind up clumping together, and that causes complications. Okay, so there are, again, antigens are anything that your immune system perceives as foreign and causes you to make a response and generate weapons like antibodies to. And these antibodies can cause clumping, like you see over here. Another term for that clumping is agglutination. Agglutination is another term for clumping. So sometimes these things that promote agglutination are also called agglutinogens. So that's another term for antigens that promote the kind of clumping you see over here. I don't typically use that word that much, but if you're reading this along in your textbook or some other sources, you may see that being used. All right, so unfortunately, if you get transfused with the wrong type of blood, this can happen. You can wind up making these antibodies that attach to those donated red blood cells and cause them to clump together. And then what happens is those red blood cells burst, they lyse, And both of those things can wind up being fatal for you if not caught in time and not treated. All right, so why would you make antibodies to somebody else's blood? Well, as I mentioned, if you looked at all the molecules on the surfaces of my red blood cells versus yours, if we had different blood types, almost all of them would be similar. There are two key types, though, that differ, and these are called your ABO antigens and your RH antigens on the surfaces of red blood cells. These can differ from person to person. All right, so let's talk about the ABO blood group first. There are 4 blood types, as you guys have probably heard before: A, B, AB, and O. And these blood types depend on particular antigens or agglutinogens, A and B, that you either do or do not have on the surfaces of your red blood cells. Okay, that's one key thing. The other thing is you make antibodies to things that are foreign to you. Antibodies are weapons that are used to combat things that are foreign to your body. So you're going to make antibodies to things that you do not have. These antibodies that you might make to these blood type antigens can also be called agglutinins, because they can be responsible for clumping or agglutinating red blood cells together. All right, so at any given time, like let's say I have type B blood, then I have B molecules or B antigens on the surfaces of my red blood cells. Okay, so I don't have A's. Type A, that's a different type, a very closely related but slightly different type of molecule that's present on the surfaces of A-type blood. So since I don't have these A molecules as part of my own body, then I am going to make anti-A antibody weapons. So those anti-A antibody weapons, if I accidentally got transfused with type A blood that has A's on the surface, those antibodies would attack that foreign type of blood and cause it to agglutinate or clump. All right, it's generally easiest to talk about this by looking at a table like this, which shows you the 4 different ABO blood types that you can possibly have. And so let's take a look at example here. All right, so if you have A-type blood, that means that your red blood cells have these A molecules on the surfaces, which incidentally are particular sugar patterns that you have on the surfaces of your red blood cells. That's what these A antigens are. Okay, so you'll have those. Those are part of you, so you make antibodies to things that are foreign to you. So you will also in your plasma have anti-B antibodies. These are antibodies that will attach to B antigens that are foreign to you. Okay. Now, what if you have type B blood over here? So if you have type B blood, then you have B antigens, molecules on the surfaces of your red blood cells. You don't have A. The A's are represented by these little blue pegs over here. So since you don't have A, in this case, you would make antibody weapons against these guys, these A molecules. Those are called anti-A antibodies. These can also be called anti-A agglutinins, because if they attach to these kinds of red blood cells, they'll cause them to clump together. All right, then finally, you've heard of people before who have type AB blood. This would be type AB. Over here, this is type A, and this is type B, and you have type AB. So notice they've got both A's and B's on the surfaces of your red blood cells. So both of those are part of you, so you're not going to make antibody weapons to either one. So you won't have anti-A or anti-B antibodies. The fourth possibility, this is called type O blood. So if you have type O blood, which many people do, you don't have A antigens or B antigens on the surfaces of your red blood cells. You're lacking both of those. So because of that, though, you make these antibody weapons against both A and B. You have both anti-A and anti-B antibodies. All right, so these antibodies over here, these are what will attack donated blood. So let's think about what will happen. Let's say you have type A blood, and let's say you receive this type over here, type B, inappropriately in a blood transfusion. Well, when that happens, your anti-B antibodies are going to attack. They will attach to those type B blood cells that have been transfused into your bloodstream, and it will cause them to clump or agglutinate, and that'll wind up blocking small blood vessels within your body, so it'll cut off the blood supply to small vessels, and then eventually your immune system comes in and causes all those clumped red blood cells to burst, which also leads to widespread inflammation across your body and causes even more problems for you. Okay, so if you have type A blood, you cannot receive B. But let's look at these other blood types over here. Let me erase all that. Let's say you have type A blood. We said we know you can't receive type B. Could you receive type AB? Could you receive this type? All right, well, let's think about it. You're type A, so you have these anti-B antibody weapons, and those can, yes, they're going to attach to those B molecules on the surfaces of these AB blood cells that have been transfused into your body. So no, you cannot receive type AB blood if you have type A, because the Bs are foreign to you and you're going to attack those foreign molecules. What about type O over here? If you have type A, could you receive type O? Yes, you could, because even though you do have these anti-B antibody weapons in your blood, there's nothing for them to attach to. The type O red blood cells don't have any B antigens on their surfaces for those antibodies to attach to. So you can receive type O. All right, let's look at another example over here. Suppose you have type O blood. This is you over here. Oops, sorry about that. Suppose you have type O blood. Okay, you can't receive blood that has any A or B antigens on its surface, because both of those are foreign to you. You have both anti-A and anti-B weapons that can attach to A's and B's. So if you have type O blood, you cannot receive A, you can't receive B, and you can't receive AB, because those antibodies have things that they can attach to on all of those different types of blood. All right, what if you have type AB blood, if we look at that for a second? If you have type AB blood, all right, so A's and B's, you have those on the surfaces of your red blood cells, so those are part of you, so you will not make antibody weapons to your own personal molecules. So in this case, what blood type can you receive? Can you receive A? Yeah, because you don't have any antibodies to attack the A's. Can you receive B? Yes, because you don't have any antibodies to attack the B's. Could you receive O? Yes, because there's nothing on the O blood to attack. So you may have heard of this before. If you are type AB blood, you're said to be the universal recipient, because you can receive any of the other blood types, A, B, AB, or O. If you're type O, you can only receive O. You'll attack every other type of blood, but you are considered to be the universal donor, because you can actually donate to each of the other blood types. Okay, so that's the ABO blood antigens are the key ones that you have to be concerned with when performing blood transfusions. All right, there's another one though. There's another blood surface molecule called the RH factor that also plays a role with blood transfusion reactions that can take place. And so we'll talk about that one on the next video lecture clip.